Welcome everybody to our ZOOMinar series. Good morning, good afternoon, and good day to all of you who are attending today. It is my pleasure to introduce Dr. Wolfgang Hoyer, who is a professor at the Institute for Physical Biology in Heinrich Heine University, Germany. Dr. Hoyer did his PhD study in the Jovin and Subramanian labs at Max Planck Institute, Germany. Then he moved to Sweden for his postdoctoral research in the heart lab at the University of Gothenburg. And he did also a second postdoc with the Convert Lab at Ruhr University, Bochum, also in Germany. His lab interest is primarily in the mechanisms of amyloid formation, with the focus of figuring out how they can modulate these processes. Welcome to our series and looking forward to your talk today.
Thank you, Magda, for this very kind introduction, and thanks to you and Rams for the invitation to this really great ZOOMinar series. I want to discuss metastable oligomers here in this talk, and the oligomers are thought to be especially critical in Alzheimer's disease. This here is the amyloid cascade hypothesis in the modified version from around 2000, where the oligomers are the first critical species, and they lead to synaptic dysfunction, neuroinflammation, and only then amyloid fibrils are formed, and tau tangles are also formed later in this process. So we have two different aggregated species here. We have metastable oligomers and amyloid fibrils. It's important that they form along different pathways. Structurally, they are related, but the difference is that the oligomers contain antiparallel beta sheets, and only the amyloid fibrils have this cross beta structure with parallel beta sheets. So the oligomers are quite difficult to capture because they are structurally heterogeneous, but this is also known for the fibrils. They can form different polymorphs, as you probably also hear later more about in Dr. Ghosh's talk. But this polymorphism is even more exaggerated here in the case of the metastable oligomers, and this is solid-state NMR data from the lab of Henrike Heise, where solid-state signals of the fibrils in blue are overlaid with those of oligomers in red. So the A-beta units within the oligomers can adopt much more conformations than in fibrils. So we have heterogeneity that makes these oligomers difficult to study. Still, it's a distinct species, and it can have specific toxic effects, of course. Because they are difficult to study, there are open questions regarding their structure and structural diversity. How and where do the oligomers form? What are their specific properties and interactions, and how do they trigger pathogenesis? This is the most important open question. So early on, it was observed that A-beta readily forms oligomers at neutral pH. In the second half of the 90s, they were termed here protofibrils by Teplow and Lansbury. So these were spherical, annular, and curvilinear fibrils. The Klein lab observed spherical fibrils and termed them ADDLs and now A-beta O's. But also those tend to arrange in a bead on a string fashion, so they also form these protofibrils, and probably these different types of oligomers form along a common pathway, and they are very, very related, although they carry different names. And because they can be readily formed in vitro, one could form these oligomers and then apply them to cell culture and found different mechanisms of toxicity, a bunch of different dysfunctions that occur then after receptor-mediated toxicity. Then we also have membrane disruption, and we have intracellular toxicity as well. So there's also intracellular A-beta toxicity by these oligomers. And all these mechanisms can explain why there's cognitive impairment then in mouse and also in non-human primate models after application of these metastable oligomers. And so this is also the reason why they are a very important drug discovery target now. And lecanemab is an antibody that specifically targets protofibrils in 2023. Results are expected for a phase 3 clinical trial, and maybe it's also directly approved because this is regarded by the FDA as a very important new therapy, and so there's a fast track application process going on. So these A-beta oligomers readily form in neutral pH buffers and can be studied, but one important observation here is that high concentration is required, high beta concentration. Usually these oligomers are prepared at 100 micromolar concentration. And then the question is are they really relevant in vivo? Because in vivo, the typical concentrations of A-beta in the brain fluids are rather 1 nanomolar or even below. So that's one astonishing observation here. And the high concentration for A-beta oligomer formation also leads to the observation that it's difficult to separate oligomer and amyloid fibril formation, because at these high concentrations, they both occur with similar rates. And we found that connecting two A-beta units on one polypeptide chain really helps in studying A-beta oligomers. We created this construct called DMAP that consists of two A-beta 40 units linked by a glycine serine-rich linker. So this leads to an increased local A-beta concentration, and this then preferentially accelerates oligomer formation and leads to nice separation of oligomer and fibril formation, as I will show you later. Initially, we tested and confirmed that this DMAP construct behaves like normal A-beta 40, so this linkage does not do anything important to the conformations that are accessible for A-beta. We first looked at the monomeric DMAP and by solution NMR, and found that the signals of monomeric A-beta 40 are retained also in this DMAP construct. There are some extra signals from glycines and serines stemming from the linker. Otherwise, the conformation is kept of free monomeric A-beta 40. DMAP can assemble into oligomers just by incubation for a short time, and then the CD signal shifts from random coil to beta sheet structure, as was observed for A-beta 40 and A-beta 42 oligomers. And also the AFM image with spherical as well as curvilinear fibrils matches the description of standard A-beta protofibrils. We also performed Cryo-EM. This was work by Mara Zielinski and Gunnar Schröder from Forschungszentrum Jülich. And they looked specifically at the smallest structures of DMAP oligomers they could observe. And this then leads to such a bowl shape 3D density reconstruction. The resolution is not nearly as high to be able to resolve anything at atomic resolution, but we can say that the density provides space for approximately 62 kilodalton of protein mass. And this would mean 6 DMAP units, so 12 A-beta subunits here. And oligomers of that size have also been described in other papers before. Then if we continue to incubate DMAP, especially under agitation, or let's say in a microplate reader, then after some time mature fibrils are formed, amyloid fibrils, and these were investigated by solid state NMR. By 2D inept mass spectra here. So these only report the mobile parts in these solids. And we have glycine and serine signals only, which means that only the linker remains flexible, but the A-beta units are both here in the core of these amyloid fibrils. So DMAP behaves like standard A-beta, but we have two A-beta units connected on one polypeptide chain. And this then leads to new kinetics of aggregation that are shown here. So by type 11T, we observe the aggregation, and we see two different regimes. In orange, we are below a threshold concentration that we call COC, critical oligomer concentration. And at this concentration, we see the typical sigmoidal aggregation kinetics that we know from nucleated polymerization of amyloid fibrils. But if we cross this COC, then we get into the blue regime here, and we gain an additional phase of aggregation. So we have biphasic kinetics. We have a lag-free initial phase, and this is highly concentration dependent here. So just by increasing the concentration of DMAP a little bit, we get a very pronounced early aggregation phase. And we resolved that by AFM and saw that in this first phase, the amyloid fibril and the oligomers are formed here from 2 to 4. And then only after the second increase in THT in the second phase, we then see the mature amyloid fibrils, and they slowly replace the protofibrils. So with this construct now, we can nicely separate the oligomer formation phase and the fibril formation phase, and can get some extra new insights on these two different phases, on their specific characteristics. In the left, we look at kinetic variability. So here are 3 replicates for each concentration. And what we observe here is that the oligomer formation phase is very nicely reproducible, but the fibril formation phase is not. So there are very different lag times here for fibril formation. In the picture on the right, we look at the dependence on the air-water interface. This is done by covering the solution with an oil layer. And so the red traces here are from the samples with the oil layer. We observe that the oligomer formation phase is not affected by the oil layer, but the fibril formation phase does not occur anymore in the samples with the oil layer. And then finally, we also looked at agitation, and I found Steve Bourgault's talk 2 weeks ago in the THT session here in the ZOOMinar very interesting, where he reported that just the movement in the plate reader accelerates the amyloid fibril formation in THT kinetics. And this is exactly what we observe here. If we prepare oligomers, if we have the oligomer phase occurring in a plate outside of the person's plate reader, the oligomers are stable for weeks. If we put them in a plate reader and just measure the sample, so without any extra agitation, we induce that the second phase here, the fibril formation phase, is occurring. So this mechanical agitation here is very important for amyloid fibril formation. So the conclusion here is that we have homogeneous oligomer formation in solution versus heterogeneous fibril nucleation at interfaces. Yeah, this is what we observe here. The next step was to look at the stabilities of oligomers versus fibrils. And for this, we use binding agents here. For Aβ, we use the ZAβ3 protein on the left that binds monomeric Aβ. And this can be used as a tool to detect monomeric Aβ, especially here this version ZAβ3W that contains a tryptophan. And when ZAβ3W binds an Aβ monomer, it shows a shift in the fluorescence emission, a blue shift from 348 nanometer to 340 nanometer. We can read out this blue shift now to say if ZAβ3W bound Aβ, so if there was any bindable Aβ in the solution. And if we add the ZAβ3W to oligomers, then we observe that the lambda max actually shifts towards the blue on a time scale from minutes to hours. So the oligomers dissolve, and the Aβ then instead goes into the binding site of the binding protein. If we add the binding protein to the fibrils, this is not happening. So here, the lambda max of the free binding protein stays the same for 7 days. So the fibrils are more stable than the oligomers. And in the next step, we then analyze if this is thermodynamic stability or kinetic stability. This is essentially the same experiment, but here we start with ZAβ3W that already has Aβ bound. We add Aβ fibril seeds, and then we observe that the lambda max increases from the bound state to the free state of ZAβ3W, meaning that the Aβ monomer dissociates from the complex and instead is built into Aβ fibrils. And these experiments then allow us to draw this thermodynamic scale here. So Aβ energy is highest in the monomer conformation, and the monomeric state, if we are above the critical oligomer concentration, of course. Then the oligomer is at lower energy. Aβ in the ZAβ3 bound state is at even lower energy, but thermodynamically most stable is Aβ within the fibril. So we can use the binding protein here to discriminate between the fibrillar state and the oligomeric state of Aβ. And we can also add the binding protein here to these biphasic aggregation kinetics. If we add the binding protein here at time 0, then we don't see any increase in thyroflavin T at all. If we add it after the oligomerization phase, then we have a complete decrease of the THT signal on the hour time scale, so the oligomers are dissolved. But if we add the binding protein only after the second jump in THT fluorescence, then we get less and less dissolution. So this indicates the fibrils are formed and they are just too stable to be dissolved here by this monomer binder. Now that we can separate oligomer and fibril formation in this construct, we can look at only the oligomer formation phase here, only at the first 5 hours. And we try to fit this to different kinetic models. On the left, we have classical nucleated polymerization, which does not give a very nice global fit here. But on the right, we have one-step oligomerization. So a simple reaction, N times molecule A makes an N-mer. This is here a global fit to this model, and there are only 2 fitting parameters. One is the rate constant of oligomerization K, and the other is N, the reaction order or the size of the oligomer. And this turns out here in this global fit to be 3.3, suggesting that we have an oligomer size of 3 DMAP molecules or 6 Aβ units. And this is a high concentration dependence if you compare it to what has been reported for amyloid fibrils. For amyloid fibrillation, the concentration dependence is usually not very high. So here for these oligomers, it's much higher. We can also look at fibril formation kinetics. This is done by subtracting the oligomer fit from the data, and then we can simply read out the lag time. Plots of lag time against protein concentrations are quite informative to get information on the mechanism of aggregation. And what we observe here is that below the COC at low concentrations, increasing concentration leads to a decrease in lag time, as would be expected. But then once the COC is crossed, we get an increase in lag time with protein concentration. And this is quite surprising, and it directly means that oligomers must actively inhibit fibril formation. This biphasic kinetics is now not something special for DMAP, an artifact of this linkage. Martin Muschol is a very important collaboration partner in this project from University of South Florida. And he provided here these biphasic kinetics of Aβ40 and Aβ42 assembly. So the same is also observed if Aβ40 is investigated here. We get the same V shape here of this T lag versus concentration plot. For Aβ42, the lag time cannot really be evaluated. And this is what I discussed before, that the oligomerization kinetics and the fibrillation kinetics overlap. They are not well separated. So this can not really be analyzed here. But what is very astonishing here is to see this really high concentration dependence of the oligomerization phase. Up to 10 micromolar, there's no oligomer formation. And then if we go to 30 or 50 micromolar, there's really a dramatic acceleration here in this oligomer formation phase. So oligomers actively inhibit fibril formation, and how do they do that? So there are different scenarios possible. For example, the oligomers might inhibit primary nucleation or secondary nucleation, so formation of new nuclei on the surface of fibrils, or they could also inhibit elongation. And we tested elongation by such an elongation assay. Here we now use Aβ40 seeds. So these are not DMAP seeds, but these are Aβ40 seeds. We add Aβ40 monomer and analyze how Aβ40 seeds are elongated by monomer in presence of different concentrations of oligomers. And the oligomers are now DMAP oligomers. And in this assay, we don't see that there's a great effect of this DMAP oligomers. In this assay, we look at secondary nucleation. This is now done by lowering the concentration of seeds so that now secondary nucleation becomes dominant, as can be seen here in these THT curves. And if we are now under such secondary nucleation conditions, then addition of the DMAP oligomers makes a difference. So apparently these oligomers inhibit secondary nucleation. And this is really dependent on the oligomeric state. So here in this experiment, we do basically the same again, but we compare 2 DMAP preparations, one in which DMAP has never crossed the COC, so it's in its monomeric state, and one where DMAP concentration was increased over the COC, so it became oligomeric, and then here it's diluted into the aggregation assay. And we see that only if DMAP went into the oligomeric state, only then we get this inhibition of secondary nucleation. What is the mechanism here? So it could be that the oligomers bind to the surface of fibrils, as is shown here in the image below, and they are covering the surface that otherwise would do secondary nucleation, and by this way, secondary nucleation could be diminished. And we tested if such binding is possible by a co-precipitation assay. We find that upon centrifugation, Aβ40 fibrils are found in the pellet, whereas these DMAP oligomers are not pelleted, but they stay in the supernatant. If we mix the Aβ40 fibrils and the DMAP oligomers, then we obtain Aβ40 fibrils decorated with oligomers. So yes, there is affinity between these two different structures, and this can explain why secondary nucleation is then diminished in the presence of these oligomers. So the outcome here is that oligomers reduce fibril formation, first by competing for the monomers, and then also actively by binding to the surface of fibrils and inhibiting secondary nucleation. And this interaction of oligomers with fibrils is interesting. If you look in the literature, it seems to be that plaque cores are surrounded by a halo of oligomers. So the idea is that you have a core made of amyloid fibrils of Aβ, and then oligomers are rather in the surroundings, forming diffuse plaques. So there's the hypothesis here on the left that plaques would buffer the oligomer concentration, but the oligomers are really the toxic species. There's some FDIR analysis here in the middle picture. There's also the hypothesis that the dense core plaques are constructed by microglia. So you have dense core plaques, and you have then a layer of oligomers and lipids around that, and the microglia are then binding to this lipid and oligomer phase. So our data now suggests that the direct interaction between fibrils and oligomers could explain why these oligomers bind to plaques. So there's no other factor needed. They have a natural affinity for one another, apparently, and this could explain these structures. Yeah, very importantly, the biphasic kinetics are not limited to Aβ, but this seems to be a general phenomenon for amyloidogenic proteins if you find proper conditions, of course. And Martin Muschel has found that lysozyme shows exactly the same behavior as DMAP here, or as Aβ. And he has published several nice papers on the different phases of lysozyme, oligomers, metastable oligomers versus fibrils. It was very nice to meet him at the FASIP conference on protein aggregation, and we looked at each other's posters and saw these plots here basically, and thought that we really have to combine this study because these proteins just behave so similarly. And also Sheena Redford had a paper on beta-2 microglobulin where she showed that there are two alternative aggregation pathways, one leading to these worm-like curvilinear fibrils and the other to the amyloid fibrils, and the one leading to amyloid fibrils is connected with a lag time, whereas the one leading to curvilinear fibrils occurs lag-free, so just the observation that we made for Aβ and for DMAP. So these metastable oligomers are formed by Aβ under physiological buffer conditions, but we have this problem with a high critical oligomer concentration. For DMAP, we need approximately 1 micromolar. For Aβ40 and Aβ42, we need tenfold or more to get into this oligomer regime. But in brain fluids, we only have 5 orders of magnitude or so lower concentrations. But there might be factors in vivo that can reduce the critical oligomer concentration. So, for example, low pH in acidic vesicles, or membranes, or post-translational modifications, or mutations, possibly. And here we looked at the low pH in acidic vesicles because it has been reported that in these acidic vesicles, in endosomes and lysosomes, there's a quite high concentration of Aβ reached, and the authors Hu et al. Of this 2009 PNAS paper calculated that there's a minimum concentration of 2.5 micromolar from fluorescence microscopy images. So we tested here for DMAP oligomers if they are taken up by neuroblastoma cells, and yes, they end up in these endolysosomal compartments here. They co-stain with lysotricha. Since it's thought that this is the place where we have the highest Aβ concentration, it's interesting now to look at the DMAP kinetics at different pH values. And at neutral pH, we have oligomer formation, we have biphasic kinetics and oligomer formation occurring on the hour time scale. And if we decrease now the value, I mean, lysosomal pH is around 4.5. So here we cover the range in the endosomal-lysosomal pathway. And at pH 4.8, oligomer formation occurs not on the hour time scale, but on the second time scale. So this is 8,000-fold acceleration upon this pH reduction. Only for oligomer formation, the fibril formation we don't observe here anymore, even if we wait for a long time at low pH. The oligomers are formed, but the second phase is lost on the time scale of this experiment. So the low pH specifically promotes oligomer formation. And these are then the AFM images of these structures. Down to pH 6.8, we observe these protofibrils. If we go down to pH 6.0, we get more and more clustering of the protofibrils, but the height of these structures is still the same, so around 4 nanometers. Then there's another change in morphology if we go below pH 6, because then they cluster into these species here that are around 50 to 100 nanometer high after drying under the AFM. And the background of the AFM substrate is clean, so we don't see these small protofibrils anymore. But since we fitted this data here, or we could fit this data globally with one reaction order of n equals 3, we think it's the same oligomerization reaction, and that just the overall morphology changes. So our suggestion is that these clusters here are really clusters of protofibrils of small oligomers. Then what about A-beta 42? Does this also form these structures at low pH? And here at pH 7, we see the typical sigmoidal kinetics of A-beta 42 aggregation, because we are below the COC of A-beta 42. And at pH 4.5, at low concentrations here, up to 5 micromolar, we still get sigmoidal kinetics. But if we go above 5 micromolar, then we have lag-free aggregation. And the AFM images support that at neutral pH, we have amyloid fibrils 10 nanometer in height. At low concentrations, at pH 4.5, we still have fibrils here, that's on the right. Again, 10 nanometers in height. But if we are in this lag-free regime, then we get 40 nanometer high assemblies, and these look like the DMAP oligomer clusters that we observed before. So it seems to be that for A-beta 42 at lysosomal pH value, we have a critical oligomer concentration of 5 micromolar, and this fits well with the literature concentration that is present in these compartments. So it's actually realistic that these structures form also in vivo. In this experiment, we then took these acidic pH-induced A-beta O clusters of A-beta 42, and we shifted them to neutral pH. And if we do that, we see that they fray out these aggregates and that these small protofibrillar aggregates are dissolving from these big clusters. And if we compare the height of these dissolved protofibrils, it's about 5 nanomolar, and this is exactly the height of de novo formed protofibrils at neutral pH. So this supports again that these clusters here formed at acidic pH are not amyloid fibril clusters or anything, but they are clusters of A-beta oligomers or protofibrils. So our suggestion now here is that the endolysosomal compartments are the prime sites for A-beta aggregation, and that here A-beta oligomer formation is especially favored. A-beta can arrive in its monomeric form, can arrive through endocytosis, for example, or it can directly be generated in endosomal-lysosomal compartments through APP processing. This also occurs in these vesicles. So there's A-beta present, and then it will form oligomers. These oligomers can leave the cell by exocytosis, but there's also more and more literature now indicating that the critical A-beta aggregation occurs intracellularly, and that this is that at the same time there is degeneration of the endolysosomal systems. So this scenario here is quite realistic, and in June there was a Nature Neuroscience paper showing that actually the amyloid plaques might originate from intracellular aggregates of A-beta that are formed in lysosomes or in degenerating lysosomes. So in the very final slides here, I want to show a connection to tau, because this is also relevant for Alzheimer's disease, and it has been observed before that tau usually is sorted axonally. So here in green, the localization of tau is given, and this is usually found in the axons. Then if Aβ oligomers are added, this leads to missorting of tau into the somatodendritic compartment, and further consequences that we have loss of mitochondria, spinal loss, and so on. So this has been shown before, and we tested this also for our DMAP oligomers, because for DMAP we cannot now make solutions that are basically 100% oligomeric without fibrils and with just low amounts of monomers left. And we see that also these solutions nicely bind dendritic spines. And then we also see that they cause tau missorting. So this is the white color here in this image down here. That is the immunostaining for tau in the somatodendritic compartment. And this is observed only for DMAP and only if it's in the neurons or in the neuroblastoma cells here in the neurons here after 24 hours incubation. So this is a very potent construct to induce this tau missorting. Yeah, now I want to conclude. I want to sum up. Metastable oligomers form a reaction that is really distinct from amyloid fibril nucleation. So they are not on pathway, but they are off pathway. The oligomers compete for monomers and inhibit fibril formation, and especially they also actively inhibit fibril formation because they interfere with secondary nucleation. So that's a direct interaction of metastable oligomers with amyloid fibril surfaces. Then acidification promotes oligomer formation drastically, and we suggest that in order to have Aβ oligomer formation of these types of metastable oligomers in vivo, you really need lysosomal conditions, because they have the high concentrations that are needed and they have the low pH that is needed for that. And finally, DMAP is a nice tool to study the activity of oligomers of Aβ, for example, the effects induced by tau missorting. I want to thank the people who have performed all the work here, especially Marie Schutzmann and Philipp Haseke, who were the main players here in these studies, and then Martin Muschol and colleagues, our collaboration partner from USF Tampa, and also our collaboration partners from Cologne and from Cambridge. And I thank you for your attention.
Hi, thank you so much for your great presentation. I'm still having a little bit of technical difficulties with my camera, I don't know. But we are ready for, I'll just take a few minutes for questions. And while we are waiting for the questions to come, I really like your idea of the oligomers being attracted to the fibrils. But I'm kind of trying to figure out how these will work spatially in the cell environment, meaning what distance or like the optimal distance for like attracting or oligomers fighting the fibrils. You understand what I'm talking about? It's just because they have to find each other. Mm-hmm. And they see each other, but what is that distances they can actually see on that?
Yeah, I mean they have to see each other in vivo then, so that this is relevant. And yeah, I showed some pictures from some papers that suggest that you have this dense plaque core, and this is thought to be composed of amyloid fibrils, and then directly around you have the corona of these Aβ oligomers. So I think the suggestion is that that really in the physiological situation that they meet. I mean, they would be direct neighbors, and they would meet one another. I mean, I think it's not really proven yet, like in these diffuse plaques and in the senile plaques, which structures are really present in there. What is the structural diversity in these different compartments? Like how do the oligomers look that you have in the diffuse plaques and so on. I think this is just not known, right? But from the gross data that we have, this directly suggests that oligomers and amyloid fibrils would meet one another, and this is in the extracellular space, right? then there's a lot of research saying that oligomers, but possibly also fibrils, are then generated first in the intracellular space. For example, there's a nice high resolution microscopy study from Aβ aggregates in lysosomes, and they found that from Kaminsky-Schierle from Cambridge, I think, and they found that in the lysosomes you really have a dense Aβ mass, and they thought it's probably fibrils, just because if you have dense Aβ mass, it's probably fibrils, but this could also be oligomers. And I think it's just not known if they can also meet intracellularly, and which species is generated.
Thank you. We have a couple more questions. One is by Rams. Great talk. Thanks. How do these oligomers differ from the micelles-like oligomers formed above CMC reported by Teplow?
Yeah, I think these are exactly the same oligomers, so they don't differ. It's the same type of oligomer.
Magdar, you are muted.
Have you looked at a covalently linked dimer with a monomer, each of Aβ40 and Aβ42? Will it help understand nature of heterooligomers formed between Aβ40 and Aβ42? And this question was posted by Bina.
Thanks. That's really a very nice suggestion. So what we struggled with a bit was to make a DMAP with two Aβ42 units, because this turned out to be quite difficult to express and purify. But finally, Marie Schutzmann managed to get this DMAP with two Aβ42 units. So that's what we have now, and it would be very nice to also have one where we have both Aβ40 and Aβ42. But we don't have that yet.
The next question is from David Welicki. Excellent talk. Do you think that the interaction of Aβ with tau is the main pathogenic mechanism, or is there another important mechanism?
Yeah, regarding to the critical toxic mechanism, I'm really very open, and it could be a direct interaction of Aβ with tau, and we are also looking into that. Yeah, I think these receptor interactions of Aβ oligomers, they seem to be very important as well and lead to many downstream effects. But I think this. Yeah, I would say I'm very open in this regard. So our study is not really giving conclusion to what is the critical toxic mechanism here.
Thank you. The next one is coming from Aphrodite. This is Q&A folder. Wolfgang, thank you for the very nice presentation. If oligomers inhibit secondary nucleation by binding to fibrils, the question is whether in the presence of fibrils, toxic oligomers get depleted.
Yeah, thanks, Ada, for the question. This could really be the case. So this is also the idea of this plaque buffering hypothesis, that oligomers are the critical species and that plaques are actually helpful because they catch the oligomers away. So this is an option, yes. Yeah, but probably hard to really prove experimentally.
And the other question is from Ashutosh. Very nice talk. Considering that aggregation kinetics is impacted by protein concentration, air-water interface, and shaking, how does one replicate what may be happening in disease state, e.g. Alzheimer's?
Yeah. Yeah, this is, of course, experimentally very difficult to find the conditions that really occur in vivo. So we see that surfaces are essential. And yeah, in vivo, there are surfaces like membranes, for example, that could play a critical role there. but it's hard to find the interface that is really important than in vivo. Shaking is increasing the contact with interfaces. So if this is then really, if any mechanical agitation is relevant in vivo, I think this is very questionable. Protein concentration is an issue, and I think what we argue here is that in the lysosomal compartments or endosomes, lysosomes, you really have high concentrations of Aβ. And this could be in vivo really the place where aggregation starts. And once the aggregates have formed, they can be quite stable. And then they might diffuse to other places, to the extracellular compartments, bind the neighboring neurons through receptors, for example, I think.
And we have two similar questions. One is from Aphrodite, and the other one is from Ghosh. The question is about whether these oligomers are toxic.
Yeah, with toxicity, I learned a lot there from my collaboration partner Hans Temple, and he thinks that these toxicity assays like MTT, for example, that they are not that important, and actually he thinks that Aβ oligomers should not have direct toxicity to neuronal cells, because this is also not observed in Alzheimer's disease, but rather they should have downstream effects, and these are then the toxic effects. So and there our DMAP oligomers and the Aβ oligomers agree in these neuronal cell cultures assay that we did, that if you apply them, you get binding to dendritic spines, you get tau missorting, for example. So these are effects that have also been observed in Alzheimer's disease, in Alzheimer's disease models, but you don't get any direct toxicity on the neurons. And according to Hans, he said that this is very good, that we don't see any direct toxicity, because this is what you would expect. And the reviewer of the paper agreed on that, and was also happy about that.
There is a question from Owie, who says, great talk, thanks. And the question is, did you manage to generate specific antibody for the oligomers?
We are trying to. We are not there yet, so we didn't yet manage, but we want to try that out. Yeah. Mm-hmm.
And there is one last question before we move to the next speaker. It's coming from MV. It's quadruple 4. It says, great talk. Is there any receptor for Aβ monomer?
I mean, there are so many receptors for Aβ reported, and now the question is, do they only all bind Aβ oligomers? I cannot really answer this question at the moment. I could imagine that some of them also bind Aβ monomers, I would guess, but I cannot really answer the question now.
Thank you so much. So if you don't mind to stay, we may get more questions at the end of the session. And we are ready to move to our second speaker. David, it's your turn. And
yeah, thanks very much, Magda. Yeah, it's my pleasure to introduce Dr. Ujjayini Ghosh. Ujjayini got her bachelor's and master's in chemical technology from University of Calcutta. And she came to Michigan State University to do a chemistry PhD. And I am very fortunate that she selected me as her PhD advisor. During her PhD, she worked on several significant projects. I think the most important was a very elegant and rigorous determination of a new structure of the hemagglutinin fusion peptide from influenza virus. So these fusion peptides are very important for catalyzing the joining of virus and target cell membranes. And they're sort of ubiquitous, where they're also important in HIV infection, as well as SARS-CoV-2 infection of cells, where SARS-CoV-2 is the pathogen of COVID-19. For the influenza fusion peptide, Ujjayini discovered a new structure that in membranes using a combination of rigorous solid-state NMR experiments and data analysis. She then went to Rob Tico's lab and did the first structures, specifically in his lab, of Cryo-EM structures of amyloid fibrils, as well as some additional solid-state NMR. She returned to my lab as a senior scientist for a couple of years, and there did a really elegant studies using relaxation NMR of lipids to demonstrate mechanistically how viral fusion peptides change the lipid molecules of membranes to predispose them to fusion. And in February of this year, she moved to an independent position at University of California, San Francisco, where she is going back, I guess, returning to doing cryo-EM structures of amyloid fibrils that are important in disease. So I think she's going to talk about her cryo-EM structure with some solid-state NMR of some different amyloid fibrils. Ujjayini.
Yeah, thank you, Dr. Velike. It was really a very nice and kind introduction. Today, I am going to talk about the work that I did in Rob Tico's lab. And for this work, actually, I got introduced to cryo-EM, and now I mostly do cryo-EM of amyloids. Some basic facts about Alzheimer's disease. Now, Alzheimer's disease has been discovered in 1906, but yet a century later, we don't know the exact mechanisms behind this disease. Several mechanisms has been proposed over the years, like the neuroinflammation, which are caused due to these amyloid fibrillar aggregates, or the disruption of cell membranes, which is caused by these oligomers, or failure of microglia to clear the waste products, oxidative damage, etc. However, A-beta aggregation is believed to be the primary cause of the Alzheimer's disease because of the mutations that are present in the A-beta peptide, as well as their precursor protein, which are associated with the familial Alzheimer's disease. So the pathological hallmarks of Alzheimer's disease is the deposition of these amyloid plaques in the brain of the Alzheimer's disease patient. However, it is not uncommon to find the amyloid plaques in the brain of non-demented elderly patients. Therefore, the connection between these amyloid plaques and the Alzheimer's disease is not very clear. So the goal of this particular work is to clarify some of the connections between the molecular structure of these amyloid plaques with the Alzheimer's disease. Now, the amyloid plaques are made of A-beta peptide, and A-beta peptide is a 38 to 42 residue peptide, which is found from a precursor protein after being cleaved by beta secretase followed by gamma secretase. The monomeric A-beta then oligomerizes, which eventually forms the fibrils. Now, these fibrils are the major component of these amyloid plaques, and that's why we are interested in these amyloid plaques. Now, amyloid fibrils are formed from a wide class of proteins, but under the EM, they look very similar, which are like filamentous protein aggregates. Although amyloid fibrils are formed from a wide class of proteins, the core of the amyloid fibrils always contains a cross-beta motif. This is a cartoon showing a cross-beta motif, where the beta strands, they run perpendicular to the fibril growth directions, and these beta strands are stabilized by the hydrogen bonds, which run parallel to the fibril growth directions. Now, the distance between these two beta strands is always 4.8 angstrom. Now, one important property of amyloid fibrils is polymorphism. It means that the fibrils that look different in the EM images will also have a different molecular structure. Now, this is a negative stain image showing 3 different polymorphs of A-beta 40 amyloid fibrils. Now, the underlying structure for this striated ribbon is this one, and for the slowly twisting fibril is this one. Now, if you look at these structures, the structures look, they're quite different from one another. But if you look at these individual subunits, they look very similar. Now, in this case, the polymorphism arises because of the different number of molecules that are present in each cross-beta layer. And as a result, each of these subunits will have a different molecular interactions, which eventually give rise to different fibril polymorphs. Now, the next obvious question is, do we see this kind of polymorphism in the brain of an actual Alzheimer's disease patient? And if yes, does it correlate with the clinical subtypes of Alzheimer's disease? So this is a very nice work done by Wei Chang. He looked at 37 different brain tissue samples from 3 different clinical subtypes of Alzheimer's disease. Typical Alzheimer's disease, or I would say it is the most commonly found clinical subtype of Alzheimer's disease, PCA variant, and rapidly progressing Alzheimer's disease. He used solid-state NMR fingerprinting to study the structural heterogeneity of these fibrils. These are the heat maps showing the difference in the 2-dimensional carbon-carbon and the 2-dimensional carbon-nitrogen NMR spectra for all of the samples. Now here the blue means that difference is the small, and where the red means the difference is larger. And as you can see, for the typical Alzheimer's disease and the PCA variant, most of this area is blue, which indicates that these patients, they do develop a single predominant Aβ40 polymorph in their brain. On the other hand, the rapidly progressing variant, they do develop at least 2 different polymorphs in their brain. So thus, we see there is a correlation between the Aβ40 polymorphism with the clinical subtype of the Alzheimer's disease. Now, the main objective of this work is to solve the structure of this predominant Aβ40 polymorph that develops in the brain of this typical Alzheimer's disease. Now, amyloid fibrils are highly insoluble and they are also non-crystallizable. I would say roughly, at least I would say 7 or 8 years back, solid-state NMR was the only method capable of giving us an atomic resolution structure of amyloid fibrils. But for solid-state NMR, you need milligram quantities of sample. But in the brain, only few nanograms of fibrils are present. And also you need to isotopically label your sample for NMR. So basically you need to amplify the fibrils. And for this, we use the self-propagation property of the amyloid fibrils. First, we purified the fibrils from the brain, and the brain homogeneous contain the Aβ40 fibrils, which acts as a seed to grow the future generation fibrils. Now, going from one generation to the next generation, we make sure that the molecular structure of the fibrils are preserved by checking their morphology in the EM images and also comparing their solid-state NMR spectrum. This is a 2-dimensional carbon-carbon solid-state NMR spectra for the first generation and the second generation fibrils. And as you can see in this spectrum, that the chemical shape for this particular label residues for the first generation and the second generation fibrils are identical, indicating that the molecular structure is preserved. This is an example of a 2D carbon-carbon and the 2D carbon-nitrogen NMR spectrum. And the first thing you will notice that the spectra is not well resolved. The peaks are broad and the spectra overall is messy. In fact, when we compare this spectra with the NMR spectra of the other Aβ polymorphs, the spectra indeed looks very messy. The cross peaks are broader, despite the fact that the fibrils are morphologically homogeneous in the EM images. So for the backbone assignment, we did a lot of 3D NMR experiments like NCA-CX experiments, where you get only the intra-residue correlations. And we also did some NCO-CX and CONCA kind of experiment where you get the correlations, inter-residue correlation. This is an example of a 3D NMR spectra. As I said earlier, for NCA-CX experiment, you only get the intra-residue correlations. And for the experiments like NCO-CX and CANCA, you get the correlation between the residue I and the previous residue I-1. And for the CONC experiment, you get the correlation between the residue I and I+1. In this way, I was able to assign roughly 68% of the sequence. Now, NMR chemical shift, it depends on the local environment. And thus, there is a correlation between the secondary chemical shift with the protein secondary structure. Now, here the negative secondary chemical shift for C-alpha and CO, and the positive secondary chemical shift for C-beta, it correlates with the beta sheet structure. And also from the chemical shift, we can get the backbone dihedral angles, as I have shown here in this plot, and they correlates well with our typical beta sheet angles. Now, the experiments that we have run so far, they all depend on the cross polarization. In a cross polarization, it transferred the polarization from proton to either carbon or nitrogen. And this magnetization transfer, it depends on the dipolar coupling. Now, dipolar coupling has an orientation dependence. What it means is that if you have a disordered segment in your sequence, those you will not see those signals in your NMR spectrum. So for this particular polymorph, we never observed any signal for the first 10 N-terminal residues, indicating that the N-terminus is disordered, and the ordered region is from K16 to V39 from NMR. Now, when you use NMR to solve the structure of an amyloid, you need an additional parameter, and that is symmetry. And we get this information from mass per length measurements. And we do the mass per length measurements using dark field electron microscopy of unstained sample. Here we compare the image intensity with the image intensity of a known fibril, known standard. In this case, this is tobacco mosaic virus. And we do this for many fibrils and plot it in a histogram. The peak of the Gaussian will give you the most probable mass per length value. For this particular polymorph, it is 28 kilodalton per nanometer. Now, A-beta-40 has a molecular weight of 4.3 kd per molecule, and the distance between the beta strands is like 4.8 angstrom. Therefore, the mass per length in this case is 3, which means that there are 3 molecules in each beta sheet repeat. Now, when you have 3 molecules, the easiest way to arrange them is that they have 3-fold symmetry like this one. Now, you cannot measure symmetry directly from NMR, but we can confirm it indirectly from the NMR. Now, if you have a structure like this one, where all the molecules are equivalent by symmetry, your NMR spectra, they will be much more cleaner, and the lines will be sharper, and you would observe a single set of chemical shift for the residues. And in addition to that, in the EM images, this kind of structure will have a rod-like morphology, because no matter how much you rotate this kind of structure, 3-fold symmetric structure, the width is always going to be the same. But for this particular polymorph, this is the negative stain image, and we can clearly see the large width modulations. Now, this width modulations, they are incompatible with the 3-fold symmetry, and it also looks like that the structure repeats itself after 180 degrees. Now, the conundrum is that you have 3 molecules in each beta sheet layer that are somehow arranged in 2-fold symmetric way. And we cannot use NMR to solve this puzzle, because NMR is based on the local structural constraints, and symmetry is a global structural constraint. And at this point, this project pretty much stalled until I got an opportunity to do a Cryo-EM on these fibrils. Now, doing Cryo-EM on amyloid fibrils is still not very easy. First, because the first is the sample preparation. Excuse me. Sample preparation. These fibrils, they do tangle easily with one another. But for Cryo-EM, you need a single fibrils. And also, in order to unambiguously unambiguously put your main chain and the side chain in your density in the cross section, you need to have at least 4 angstrom resolution or better than that. And finally, the image analysis for this particular data set was very challenging because of the low molecular weight of A-beta combined with the large width modulations. So this is an example of a Cryo-EM image of this polymorph, and you can clearly see the width modulations that are present here, which means that these width modulations are intrinsic to the fibril morphology. I want you to focus on this area. And as you can see that the signal to noise, particularly in the wider section, it is very poor. And it actually caused a huge trouble in our image alignment. Now for amyloids, you do helical reconstruction. You can imagine the fibril as an helix where the subunits are regularly arranged along the axis of the helix. Now the helix is defined by two parameters: helical twist, which is the angle between the two subunits, and helical rise, which is the distance between the two subunits, and it is parallel to the helical axis. Now, unlike solid-state NMR, Cryo-EM data analysis is completely different. Here you go from the global structure to the local structure. The Cryo-EM image is basically a 2D projected image of a 3D object. Since the contrast in this image is very low, you need to average over a huge number of fibril segments to get a high-resolution 3D structure. First, you slice individual fibrils into smaller fragments. They are called particles. And these low signal to noise particles, they are clustered together and averaged to get high resolution 2D classes. These are an example of 2D classes. And here you can clearly see the beta strand separations. Now, these 2D classes are further taken into 3D classification to get a density. One of the main purposes of 3D classification is to further subdivide your data set into more homogeneous data set. In this case, I have 3 different classes, and as you can see here, and they have different number of particles, different helical parameters, and the resolutions, they're also different. Since class 1 has the highest number of particles and the best resolution, we further refine this class. This is the density of reconstructed density along the fibril growth direction, and we can clearly see the beta strand separation, and this is the cross section. Now, although the resolution was 4.5 angstrom, we couldn't place the main chain and the side chain into this density unambiguously. And we tried different things, but it didn't work out. It didn't work out. So finally, we looked into the Reliance software that we are using. Now, as I mentioned earlier, that the signal to noise in the wider section, it was very low. So, and as a result, we do have a lot of trouble with the image alignment. But one of the major advantage of helical symmetry is that the orientational parameters of these fibril segments that come from a single fibril, they are fixed relative to the helical axis. So if you know the orientation parameter of one of these boxes, you can make a reasonable case for the other ones. I'm not going into details about this slide, but I want to say that in Cryo-EM, if you want to get a high resolution structure, you need to precisely align your particles. The better your alignment is, your structure is going to give you a higher resolution. And for Reline, it aligns the particle using 5 variables, 2 positions, and 3 angles. We found out that the standard Reline uses the correlation in the psi angle from one box to another, but it does not use the correlation in the rotational angle. This is something we had to implement. And this work was done by Kent. He implemented all these orientational correlation in the modified Reline version that we used. Apart from the orientational correlation, Reline also has a trouble in aligning particles. Now, these fibrils, they have a directionality. Going forward versus backward, they are different. And we observed it, we noticed it in many of our runs. Here, I want you to focus on this number, which is 38.346%. It means that 38% of the fibril segments are aligned in the wrong direction. So after doing extensive alignment and after the implementation of the rotational correlation, we were able to improve the alignment, precision of the alignment by almost 90%. This is the final reconstructed density, and you can clearly see the difference. The density does have a 2-1 screw symmetry, and it looks like there are 2 complete molecules, which are the central layers, and the third molecule is somehow chopped into 2 halves, and thus accounting for a mass per length of 3, despite of having a 2-fold symmetry. So first I'm going to talk about the central layers, and then I will come back to these additional layers. So the density of the central layers spans from residues 10 to 40, and the N terminus was disordered enough, so we didn't get any density for the N terminus, which is consistent with our NMR data. In the next step, we combine the Cryo-EM data with the solid-state NMR data in Explore NIH software, and this is the final refined model. So in the refined model, both these protofilaments, they are fully extended. Thus, N terminus has a stretch of beta sheet from residue 12 to residue 22, and the C terminus has stretches of 3 short beta sheet. And the N and the C terminus, they are separated by this turn region as is shown here. And the turn is mostly stabilized by an electrostatic interaction. And the protofilaments are stabilized by the hydrophobic interactions. Next, we probe the hydrophobic interaction by solid-state NMR. So what we did is we selectively excited the aromatic ring of the phenylalanine and watched the signal grow as a function of mixing time. Now, at longer mixing time, you can see that the signals from alanine, valine, leucine, isoleucine, they pop up, which were absent in the 50 millisecond spectrum, which means that all these hydrophobic residues, they are in contact with this or interacting with this F19. As I said earlier, the N and the C terminus, they are separated by a turn region. And we probe the electrostatic interaction between D23 and K28 using Reader solid-state NMR experiment. In Reader, the dephasing is depends on the dipolar coupling, and dipolar coupling has a inverse cube distance dependence. So therefore, when the distance is shorter, we would expect a substantial Reader buildup like this one, and when the distance is longer, the Reader dephasing buildup will be much more flatter. So this is our experimental data, and it matches with a 4 angstrom distance, thus confirming the presence of a Sol bridge between D23 and K28. Now, what about the additional densities? Now, the mass per length for this polymorph is 28. The mass per length of this individual layer is 9 kilodalton, and 2 will account for 18 kilodalton per nanometer. Therefore, the remaining 10 kilodalton should come from these outer layers, and each of them should contribute 5 kilodalton per nanometer each. Now, from the overall density, it seems like these are also cross beta motif, and there is a beta sheet which is like 8 or 9 residues in long, and we can see a clear helical, beta strand separation along the helical growth axis. We thought about several possibilities, but we ruled out eventually because they were not consistent with our data. And finally, we came up with this beta hairpin idea. So beta hairpins are also cross beta motifs, but here they are anti-parallel cross beta motif. So the repeat distance is 9.6 angstrom. Therefore, the MPL of each unit is 4.5 kilodalton per nanometer, and 2 subunit will account for the remaining 10 kilodalton per nanometer. And we came up with this kind of model where you have 2 central layers, and the outer layers are beta hairpins that span from one layer to the next layer. This model is consistent with our mass per length data and also our solid-state NMR data. So we tried to refine our structure with 9.6 angstrom helical rise, but it didn't improve our resolution. And it makes sense because these beta strands, they are made of different residues. So every time you will end up getting a superposition of these 2 densities. But surprisingly, we do see a weak 9.8 feature in the 2D Fourier transform of the fibrils from our Cryo-EM images. And this is consistent with, this intensity is consistent with the 2D Fourier transform of one of our models that like this one, simulated models like this one. In the next step, we actually used NMR to see if we do have beta hairpins or not. And for this, we have these anatomic models. Here, the beta strands are from residue 16 to 22, and another stretch of beta strand is from residue 30 to 36. The highlighted region is the turn region. Then we came up with two different labeling schemes. If we have a beta hairpins, the shortest C13 and N15 distance should be in the range 4.5 to 6 angstrom. And if it is a cross beta unit, the shortest C13 and N15 distance would be either 6.5 or 8.5 angstrom. Then again, we used REDOT to probe this distance. And as you can see, for both the peptides, our distance matches with the 5 angstrom, which kind of confirms that, solid state, confirms the presence of solid beta hairpins. This is the final structure. Sorry. This is the final structure. Now the central layers there are two central layers. Those are fully extended, and these two protofilaments are stabilized by hydrophobic interaction and also electrostatic interaction. The outer layers are beta hairpins that spans from one layer to the next layer. Now this is the first structure that shows the presence of both parallel and anti-parallel cross beta structure in a single structure. Now, there are some novel aspects of this work. First is we use seeding to grow the second generation fibrils that are sufficiently clean for Cryo-EM. And as you can see here, for the first generation fibrils, a lot of materials are stuck, brain materials are stuck to the fibrils. So in this case, the image analysis would be very difficult, but the second generation fibrils are much more cleaner, which helped image analysis. Also, we implemented the rotational correlation in the Reliance software, which which is available in the current version of the Reliance. And hopefully today I was able to convince you that Cryo-EM and solid-state NMR are highly complementary techniques. Now here I have shown all the structures of the A-beta fibril polymorphs that have been solved so far. And you can see the structures are very different from one another. But all these structures, they have a common motif, like a U shape or S shape or C shaped motif. However, the structure that we solved is in a fully extended conformation. And this is the first time we have reported a fully extended conformation for beta amyloid assemblies. Now, the existence of beta hairpins in amyloid fibrils, they have been reported in MD studies. However, they hasn't been observed experimentally. And this is the first time we observed the existence of beta hairpins, and we have we observed the beta hairpins experimentally. Now, this structure is completely unanticipated because there are two different structures present in a single structure, and this is not anticipated because the fibrils look extremely homogeneous in the EM images. And with that, these are the list of the NMR experiments and the additional experiments that we have to do for this polymorph to get the structures. And finally, I would like to thank Dr. Tico. He encouraged me and honestly pushed me to learn Cryo-EM, and that's really very, very good. And Kent, who did all the code, who wrote all the code and who did all the implementation in the Reliance software, and we had to work very hard for to get this structure. Ming, he prepared all the peptides, and also at that time my group members, they're very nice, and also I often go and talk to Jenny and Bartram when I had any issue, and then Huimin helped me to get the data in Krios, and Haifeng trained me with the train me how to freeze grids. And with that, thank you all, and I'm happy to take any questions.
Yeah, great talk, Ujjayini.
Thank you.
Let's see, I guess, so yeah, we don't have any questions yet. So did you, or I guess anybody in Tico's group, try at all to remove the sort of outer layer, the beta hairpins, and by some chemical method or pH or?
I don't think so, actually. Yeah.
I mean, I don't know if maybe it's just visual. It sort of looks like in your structure that they are, the binding would be less. I guess probably it's the hydrogen bonding, right? There's not, there's fewer hydrogen bonds. Is that fair to say, or is that not true?
Sorry, what was the question, Dr. Maliki?
Just that it looks like the beta hairpins have fewer hydrogen bonds with the core than the core has with itself. Is that?
Yeah, it could be like, we cannot rule out the possibility that it might be attached to the central layers much more loosely. It could be possible, but we don't know. I cannot tell you for sure. Yeah.
Let's see. I'll ask you another, I know this is maybe a little nitpicky solid-state NMR question, but I think on your Saltbridge NMR data, right, it's not really a great fit. The read-or data don't fit that great. And so it looks like maybe there's actually two different structures there. You may not have, well, I don't know, did you think about that at all?
Yeah, it is possible, like, some of them, they do have like a little bit fluctuation. It's an average bulk distance, so it is possible. Like instead of like 4.1 angstrom, the average distance might be like 4.2 or 4.3, but it is possible, yeah.
No, it's more systematic. It's that at kind of earlier, shorter dephasing times, right? Your experimental dephasing is higher, and then at longer dephasing times, your experimental dephasing is lower. So that, seems to me at least one possibility. You have two populations of structures, one with somewhat shorter distance and one with a somewhat longer distance. Anyway, yeah. Okay, so we have a question from Rams. Great talk, Ujjayini. Could you comment on the stabilities of your fibril structure with other reported structures of A-beta?
Yeah, sure. So the central layers, I would say the inner layers, they are very stable. But as far as the outer layer goes, like they're always there, and we think that it is a part of the structure, but there is a possibility they might be like loosely attached to the structure. So they might not be as stable as like the central layers. So that's one thing I think. Yep.
Very good. And the next question is from David Eisenberg. Do you observe any A-beta oligomers in your preparations?
No, actually, I did not observe any A-beta aggregates. No, it's just the fibrils. Yep.
So no oligomers.
No aggregates.
Yeah. Next question is from Ashutosh Tiwari. Very nice talk. Have you compared the NMR structure of A-beta with the Cryo-EM structure? What are the similarities and differences? I guess that's, would say, did you actually do a de novo NMR structure of your preparation?
No, we couldn't do that because of the symmetry issue. That's why we have to go to Cryo-EM, because we didn't know how to place the third molecule around the helical axis. So we couldn't do NMR structure determination. But in the final structure, we use the NMR structural constants.
Right, if I understand it right, you sort of the core, whatever, 2/3 of the fibril or molecules in the inner core, that's the Cryo-EM. And then the beta hairpin, is that really a combination of Cryo-EM and NMR? I mean, do you actually fit that cryo-EM electron density?
Yeah, we did fit the cryo-EM electron density, but for that, we didn't have any NMR structural constraints. Because,
I see.
because in the NMR, we do see two different sets of chemical shift, and one is like very sharp, we assigned it, but the other one, which is like a broader, we couldn't assign it. So it is possible that it is most likely that they're coming from those the beta hairpins. And beta hairpins can have different orientations, like it could be like this, it could be like this, or it could be like this. So we actually don't know which one is there, but other than that, there is a beta hairpin in there.
So the next question is from S. Becker 2. Could the outer layers be something else than protein?
We don't think so, because we have done mass per length several times, and they always came out to be 3. So that's why it should be protein. If it's not there, then we would expect a mass per length of 2.
Sure. I guess this was sort of getting back to kind of just a little bit of the earlier discussion. And can you fit actually side chains, kind of with the right sequence into the outer layers? Is your resolution of the cryo-EM, enough for that?
No, they are not. As you can see here, they are not. We always get the superposition of the 2 densities. Maybe sometimes it fades, but it's not always exact, like the central layers.
So it's kind of, it's right. So you're saying it's from sort of the supporting measurements rather than the electron density, that you're saying that it's protein rather than some other chemical compound. So great. It looks like that is all the questions. So I guess I hand it over to Rams. Oh,
great talk, Ujjayini, and
just one quick one more.
Appreciate it. So I have a question, quick question.
Okay.
So you said the outer layer can be slightly less stable than other structures. Do you think this structure can convert into other regular fibril structure as a function of time, or maybe heat, or any other treatment?
Can turn out. So I think that the central layers are very stable, but for the outer layers, there is a possibility that, like when the fibrils are formed, new Aβ, like the new molecules get attached, and somehow they form fibrils, and they move on, and then they get to the next one, then the next one forms. So something like that, there is a possibility. But the one thing I want to mention that Rob did mass per length at several time points, but he always observed that the mass per length is 3, which means that these are always there. It's not like a secondary nucleation, which can just easily goes off and come back. So that is also another thing. But to me, it looks like these structures might be less tightly bound than the central ones. And maybe it is there, there are some hydrophobic groups around this region. So maybe since to patch to cover the hydrophobic groups, the beta hairpins are present there. So that's my thinking.
And do you see any possibility for water channels?
Like this one, water could go maybe, but we didn't check it.
There is a question in Q&A from Ada.
Yeah, for Aphrodite Kapuniotu. Do you know something about the structure of the seeds which were used to generate these fibrils?
Yeah, so for the seeds, so we made sure when we used the seeds, we looked at the morphology of the seeds in the EM before we started the seeding. And after seeding, the morphology of the fibrils, they look same, like the weight, the crossover distances, they are same. So that's why we suspect that the structure is same. But yeah. So we check the morphology. But we cannot directly do like NMR on the seeds because we need C13 and N15 levels in any case. And for, yeah, definitely you can do cryo-EM. Nowadays, like we do cryo-EM directly on those brain-derived materials without seeding, but yeah. But I haven't checked it.
I guess we can open up for both the speakers. Wolfgang, are you around? Okay.
Yes, I am.
Great talk. I wanted to ask you another question on the micelle like oligomers that Teplo reported. Those have critical pH as well, critical temperature as well, in addition to critical concentration. So the fact that you see different kind of clustering with pH, what do you think the role of critical pH? Do you think these oligomers are same in secondary structure and the intermolecular interaction at different pH? Could you comment on that?
Well, one thing I can comment is that we see that if we reduce the pH, the critical oligomeric concentration is reduced. So we get into the range of 0.1 micromolar, and then we cannot really get a good THT signal anymore. So we don't know how low the COC is really at the low pH. Structural information and independence of pH, we cannot provide. We don't have structural information. I mean, the AFM is what we see, right? And there is certainly clustering occurring on the higher length scales, but I could imagine that the structures, that the molecular structures themselves, that they are rather similar at different pH values, but this remains to be proven.